Okay, one, two, three, go. Good day, everyone. Today, our group will discuss the genetic of cancer, a topic that focuses on how changes in our genes contribute to the development and progression of cancer. Cancer is not just one disease, it is the result of complex interactions between genetic mutations and environmental factors. We will look at how mutations in specific genes, such as DNA repair genes, genes regulating telomerase, and genes that promote vascularization, can disrupt normal cell functions and lead to uncontrolled growth. We will also explore how virus infections can play a role in cancer development. Through this discussion, we aim to better understand the genetic mechanisms behind cancer and why studying these processes is important in finding more effective treatments and prevention strategies. Next slide. So, our topics, the list of key concepts are DNA repair genes, genes that regulate telomerase, genes that promote vascularization, and lastly, virus infections and cancer.
All right. Next slide, please. So, the integrity of DNA is under constant assault from radiation, chemical mutagens, and spontaneously arising changes. In spite of these damaging agents, the rate of mutation remains remarkably low, thanks to the efficiency with which DNA is repaired. So, what is DNA repair genes? Next slide, please. So there are a number of complex pathways for repairing DNA, but several general statements can be made about DNA repair. First is that most DNA repair mechanisms require two nucleotide strands of DNA. It is because most replace whole nucleotides and a template strand is needed to specify the base sequence. A second general feature of DNA repair is that is redundancy, meaning that many types of DNA damage can be corrected by more than one pathway of repair. This redundancy illustrates the extreme importance of DNA repair to the survival of the cell. If a mistake escapes one repair system, it's likely to be repaired by another system, ensuring that almost all mistakes are corrected. Now, we talk about different the DNA repair. So, what are the different types of DNA repair? Next, please. So, the first type of DNA repair is mismatch repair. So, before we will talk about this, let's ask a question that does proofreading eliminate all errors made during replication? Well, the answer with that one is no. So while proofreading significantly reduces the error rate, not all mistakes are fixed on the fly by DNA polymerases. What mechanism e- exists to correct the replication errors that are missed by the proofreading function of DNA polymerases? So this is mismatch pair. So errors that slips by proofreading during replication can be corrected by a mechanism called mismatch repair. So while the error rate of DNA replication is about 1 in a 10 to the power of 7 nucleotides in absence of mismatch repair, this rate is further reduced into 100-fold to 1 when mismatch repair is functional. So what are the basic tasks of the mismatch repair system? So first, it scans newly made DNA to see if there are mispaired bases. Like if G is correctly paired to a T, which is we know that in the complementary bases, G is complement with C, and T is complement with G. Yeah. So next is it identify and cut out region of the mismatch. Then lastly, it correctly fill in the gap created by the existence of the mismatch region. So this involves for a while. This involves proteins such as mute S, mute L, endonuclease mute H, exonuclease, helicase, DNA polymerase, and ligase. So what are the steps or the mechanism of the mismatch pair? So first, the mute S and mis recognizes mismatch and binds with it. So if there are incorrect match between the GATC, it recognizes, and then the mute L will complex with mute S, and then mute L activates endonuclease mute H, which uh, which this will create a loop within that that strand or the daughter strand. Then the exonuclease and helicase, the exonuclease and helicase excise portion of daughter strand. Then lastly, the DNA polymerase 1 or DNA polymerase 3 fills the gap, which will then seals off by the ligase. Next type of DNA repair is direct repair. Next slide, please. So, what is direct repair? Direct repair does not replace altered nucleotides, but instead, it restores their original or correct structures. So, there are two examples of DNA damage that can be reversed, which are UV induced lesions and alkylated bases. So, cells may possess mechanisms to repair types of damage that occur frequently. For example, there are proteins that remove alkyl groups from DNA bases. So, most organisms, especially those exposed to high amounts of UV radiation, have enzymes that can directly repair thymine dimers. The process called photoreactivation or light repair uses an enzyme photolyase that depends on visible light to directly break the bond joining the thymine bases. So there are two types of direct repair. Next slide, please. So first is the photoreactivation. So what is photoreactivation? It is an enzymatic cleavage of thymine dimers activated by visible light which is only present in the prokaryotes. So it is a UV-induced lesions caused by UV light that can be reversed through a process called photoreactivation, which uses visible ener- light energy to break the damaged DNA structure, restoring the original pyrimidine bases. So what is the mechanism of the photoreactivation? So first, so as you can see in the image or in the figure, enzyme photolyase encoded by PHR gene binds to a pyrimidine dimer. Then visible light shines on cell, which then FADH absorbs that light and releases electron. Then the electron interact with the pyrimidine dimer, which then splitting the cyclobutane ring in dimer due to the electron interaction. And then finally, enzyme leaves the DNA and the DNA structure returned to its prior state. Next type of um, direct repair is the removal of methyl groups. Next slide, please. So, in removal of methyl group, alkylated bases can be reversed by enzymes such as O6-alkyl guanine DNA alkyl transferase or the IGT and ALKB related dioxygenases, which removes or modifies the alkyl group respectively. So, what is the mechanism of the removal of methyl groups? Basically, so as you can see in the figure, enzyme 06-meth ileoguanine, or the methyl transferase encoded by the ADA gene in E. coli. So this is an example of an E. coli, recognizes 06-methylguanine in duplex DNA, which is as you can see there. Then it removes the methyl group, transferring it into an amino acid of the enzyme. Then the methylated enzyme is no longer active. Hence, this has been referred to as a suicide mechanism for the enzyme. Next type of DNA repair is the excision repair. So, excision repair is a general mechanism of DNA repair that various enzymes are involved that can sense DNA damage. So, excision repair is categorized into two parts: base excision repair, or the BER, and nucleotide excision repair, or the NER. So, let's start first with the BER, or the base excision repair. Next slide, please. So, base from a nucleotide within DNA can be removed in several ways, such as radiation, by spontaneous hydrolysis, by an attack of oxygen free radicals, or by an enzyme like DNA glycosylase. So, BAR repairs DNA bases damaged by alkylation, deamination, oxidation, and lost bases. So, what is the mechanism of base excision repair? By using an enzyme. So, in based on the graph, or like based on the figure, the uracil DNA glycosylase rather enzyme recognizes uracil within the DNA and cleavages it out at the base sugar bond, which is a glycosidic bond. The resulting site is called AP, or the apurinic aprimidi, uh, apurinic pyrimidinic site because of lack of purine and pyrimidine. The AP site, AP endonucleases, then sends the minor distortion of the DNA double helix and initiate the excision of single AP nucleotide. The AP endonucleases class 1 nick at the 3' side of AP site, and the class 2 nick at the 5' side of the AP site. The DNA polymerase then inserts a nucleotide at the AP site. The lyase, or the phosphodiesterase, then removes the base free nucleotides. Then the DNA ligase close the nick. So, the other type of excision repair is nucleotide excision repair, or the NER. So NER pathway can recognize and remove a wide variety of bulky helix distorting lesions from DNA. It repairs DNA damage which are produced by the UV component of sunlight. While mechanistically similar to BER, the NER pathway is more complex, requiring some 30 different proteins to carry out a multi-step cut and patch like mechanism. So the biological importance of NER is that defects in NER cause several human genetic disorders, including Xeroderma pigmentosum, Cockayne syndrome, and the trichothiodystrophy, which are characterized by extreme sun sensitivity. The general mechanism of a nucleotide excision repair is that the ABC exonuclease, which are composed of subunits encoded by UVRA, UVRB, and UVRC genes move along DNA and can detect thymine dimers and 4 excision endonuclease. The UVRA and UVRB complex attach on distortion site. Then UVRA will dissociate it. The UVRB attracts UVRC and nicks 5 nucleotides at the 3' side of DNA, while 8 nucleotides nicks at the 5' side of DNA will be produced by the UVRC subunit. The UVRD or DNA helicase 2 removes 12 oligonucleotides. Then, the DNA polymerase 1 now fills in the gap in the 5' to 3' direction. After the filling of the gap, the DNA ligase seals that gap. So that is the the DNA repair genes. Moving forward to the next topic.
Thank you for that, Lawrence. Now, moving forward, let's talk about the topic genes that regulate telomerase. So, this part is very important because telomerase is one of the main reasons cancer cells can divide endlessly. By understanding these genes, we can see why cancer is so hard to control, but also why telomerase is such a good target for therapy. So, let's start with the first topic. Next slide, please. Background on telomeres and telomerase. So, telomeres, as what we all know, are repetitive DNA sequences, specifically the TTAGGG in humans, at the ends of chromosomes that protect genetic material during replication. With every cell division, telomeres shorten, eventually leading to chromosomal instability, cell cycle arrest, or apoptosis. Apop- a- apoptosis, sorry for that. So, telomeres are repetitive DNA sequences, and it is located at the ends of our chromosomes, and their job is to protect the chromosome ends from being damaged or mistaken as broken DNA. So we can think of them as the plastic cups on shoelaces that prevents fraying. So every time our cells divide, DNA replication cannot fully copy the very end of our chromosome. And because of this, telomeres gradually get shorter with each cell division. And when they become too short, the cell senses danger and either stops dividing, that is what we call senescence, or it can also undergo programmed cell death, which we call apoptosis. And this is actually a natural safeguard against uncontrolled cell growth. Now, on the other hand, we have here telomerase. Telomerase from the ending ASE is an enzyme. Yes, it is an enzyme. It is a reverse transcriptase that adds telomeric DNA repeats to chromosome ends, maintaining length and stability. So telomerase is simply the one responsible for regulating the ends of our telomeres, adding the sequence TTAGG, maintaining length and stability of our chromosomes. Next slide, please. So this is how telomeres and telomerase looks like. So here we have our chromosomes, and at the end we have the blue one. It is like the cap. That is the telomere, and on our telomeres we have the sequences, the TTAGGG, and the telomerase, the orange one. They're the ones responsible for maintaining that sequence. And sa kilid niya we can see the different molecules helping the telomerase in regulating our telomeres. We have the discarine, GAR, NHP2, and OP10. These are molecules or specifically proteins. So normally, telomerase are active in germ cells, stem cells, and some immune cells, but it is not active in most normal body cells. It's usually restricted to germ cells, which gives rise to sperm and egg, stem cells, which needs to divide many times to repair tissues, and some immune cells. And for most somatic cells, telomerase stays switched off. And this is why our body cells have a natural limit to how many times they can divide. So, unsa may possible? What are the possible situations that might arise in this if our telomerase is usually switched on, right? So we have here, next slide please. The role in cancer progression. So yes, telomerase have a role in cancer progression. So in many cancer cells, the genes regulating telomerase expression are mutated or deregulated, which leads to inappropriate activation of the enzyme. So we have here common examples. As we can see in the picture, we have HDART promoter. So on the following slides, we will explain this deeper, but let's have the gist here. So as we can see, HDART is actually a viral agent or an adenovirus, and it we can see here the comparison between cancer cell and normal cell. So in the normal cell, telomerase is inactive. In cancer cell, telomerase is active. If the telomerase is active, there will be viral replication of the adenovirus, which results to cell destruction, viral release, and virus spread. In contrast, if the telomerase is inactive, especially in normal cell, there will be viral replication blockage. Next slide, please. Excuse me. So another example is TERT functions in cancer formation. So TERT, which will be explained also later, has a role in cancer progression. So it can be in transcriptional regulations, or telomere maintenance. So in transcriptional regulation, TERT promotes mutation. How? It influences telomerase activity, assembly, and also recruits telomerase to telomere. And with that, it became a therapeutic target for small molecular inhibitors or TERT vaccine. We have here the upstream and downstream. For upstream, we have post-translational modifications, we have phosphorylation, ubiquitination, sumulation. And for not downstream, we have the non-canonical functions, proliferation, apoptosis, cell cycle, tumor initiating cell formation, EMT, induce cancer cell invasion, and influence transcriptional factors activity. Next slide, please. So, for a while. Sorry for that. So, moving forward for, let's gonna dig deep, no, in detail for the genes that regulate telomerase on the progression of how telomerase in affects cancer. So, let's dig into the genes and proteins that directly regulate telomerase. And first, we have the TERT. This is the one I've constantly mentioned earlier. It is the telomerase reverse transcriptase. This is referred to as the engine of the enzyme telomerase, which encodes the catalytic subunit of telomerase, and it is normally silenced in somatic cells. So, what is its effect in cancer? It allows continuous cell division, which results to immortalization of cancer cells, which is usually seen in cancers such as glioblastoma, melanoma, bladder, and thyroid cancer. Next slide, please. For the second one, we have TRC, or the telomerase RNA component. So, telomerase is not just a protein, although it is an enzyme, but it is not just a protein. It also needs an RNA template, and TERC, it is the one responsible for providing this RNA sequence. When I say RNA sequence, I'm talking about the TTAGGG repeats, which telomerase copies to elongate the telomeres. And without TERC, telomerase cannot function at all. And mutations in TERC can cause premature aging syndromes like dyskeratosis congenita, but in cancer, the availability of TERC ensures that telomerase keeps extending telomeres, feeding tumor growth. Next slide, please. For the third one, these are the regulators. We have descarin, DKC1, and associated proteins, NOP10, NHP2, and GAR1. So these stabilize DRC and assemble the telomerase complex. So these proteins are like the construction crew, and they stabilize TRC, which helps assemble the complexity of the telomerase. So in normal cells, if discarin is defective, telomerase becomes unstable, which leads to, yes, premature aging. But in cancer, tumors often overexpresses these proteins, which keeps telomerase stable and fully functional, which supports uncontrolled proliferation. Next slide, please. For the fourth one, we have MYC oncogene. So, transcription factor that directly activates TRT gene expression. So MYC is a powerful transcription factor often called the master switch of growth. So why is it called master switch? It is because it can directly bind to the TERT promoter and activate its expression. So it is on MYC that has the ability to either switch it on or switch it off. And in many tumors, MYC is overexpressed, which means it is constantly switching telomerase on. And this is one of the ways cancer ensures it never runs out of telomeres. Okay, just like what I've constantly said, if telomerases or telomeres are turned on, then cancer they are given the chance to continuously reproduce. That's how it kind of happens. And for the fifth one, we have tumor suppressor genes, specifically the P53 and RB. So P53 and RB, or RB retinoblastoma protein, it normally repress TRT expression. So they suppress TRT and keep telomerase off in somatic cells. But in many cancers, P53 and RB are mutated or inactivated. And when the brakes fail, telomerase activity shoots up, contributing to uncontrolled growth. So tumor suppressor genes, from the term suppressor, it suppresses. So they basically act like as the brakes. If these brakes cannot function well, then it contributes to uncontrolled growth in cancer cells. That's how it. That's the gist, guys. Next slide, please. For the sixth one, we have epigenetic regulators. So the DNA methylation and histone modifications in the TRT promoter affects whether it is turned on or off. Example: TRT promoter methylation is often linked with cancers that show telomerase activation. So in this one, um, histone modifications can also make the TRT gene more accessible, and these epigenetic changes act like hidden switches that flip telomerase back on in tumors. And with all that being said, let's have a summary. Next slide, please. All right, so I know that some of the terms a while ago are very deep, but let's just have the gist here. So mutations in the promoter region of the TRT gene, or the telomerase reverse transcriptase, it leads to abnormally high transcription, which I said earlier, common in glioblastoma, melanoma, bladder cancer. Epigenetic changes, specifically DNA methylation, histone modification, can also activate telomerase genes. Oncogenes, specifically MYC, stimulate telomerase expression, and tumor suppressors, specifically P53 and RB, normally suppress telomerase, and their inactivation indirectly promotes telomerase activity. Next slide, please. Another summary. So, what are the consequences, again, of telomerase activation from what I've said earlier? Immortalization of tumor cells. Yes, it leads to indefinite proliferation. Genome instability is reduced because telomeres don't shorten to dangerous levels. Cancer stem cell maintenance. Telomerase helps preserve a pool of immortal cancer initiating cells, and tumor progression and metastasis. By preventing senescence and apoptosis, tumors can grow aggressively. Thank you. Next slide, please. All right, so since we're done talking about the genes that regulate telomerase, let us now talk about how to target telomerase in cancer regulation. So, telomerase is a prime anti-cancer target because it's inactive in most somatic cells, but abnormally reactivated in approximately 90% of cancers. By selectively inhibiting telomerase activity, cancer cells can be driven into senescence or apoptosis while sparing most normal cells. Next slide, please. So, the most common therapeutic strategy is, first we have telomerase gene therapy. So, for its concept, for a while, sorry for that. It uses vectors to deliver genes that selectively kill telomerase positive cells or cancer stem cells while leaving telomerase negative cells, the normal somatic cells, unharmed. So, how does this work? Scientists design gene vectors that place a suicide gene under the TERT promoter. And since TERT is active in cancer but silent in most somatic cells, only the cancer cells activate the suicide gene and die. So, a telomerase gene therapy vector, specifically TERT promoter driven suicide gene, when it is introduced, the in normal stem cells, the therapy has no effect, so they survive. But in cancer stem cells, the one that telomerase is positive, the suicide gene is activated, which results to cell death. Next slide, please. Next slide, please. All right. So second one is suicide gene therapy or the prodrug activating strategy. So for its concept, it introduced an enzyme encoding gene into cancer cells under control of the telomerase promoter. So cancer cells express the enzyme. This is how it works. A prodrug or a harmless compound is administered. Inside the cancer cell, the enzyme converts the prodrug into a toxic metabolite or a toxin, and that toxin kills the cancer cell and can spread to neighboring cells, which is what we call the bystander effects. So, in other terms, no, in simple terms, when the cancer cell activates a harmless prodrug into a um, toxin using an enzyme controlled by telomerase promoter, this not only kills the cancer cell but it can also affect the neighboring cells. And that is how bystander effect happens. Next slide, please. Next one is oncolytic virus therapy. So, engineer oncolytic viruses that replicate selectively in telomerase positive cells. So, for its mechanism, in normal cells, replication is abortive, which means virus fails to reproduce. While in cancer cells, virus replicates actively, which results to cell lysis and then results to new viruses, which infect neighboring cancer cells. So, the outcome of this cancer cells selective killing and amplification of the therapeutic effect. So in other terms, since viruses are engineered to replicate only in telomerase positive cancer cells, normal cells do not support viral replication. But in cancer, the virus multiplies, which bursts the cell and spreads to infect others. Next is direct telomerase inhibitors. So the best example is imetelstat or the GRN163L, an oligonucleotide that directly binds to the RNA template or the TRC, preventing telomere elongation. So it blocks telomere elongation and it's already in clinical trials for blood cancers like myelofibrosis. Yes. Next slide, please. We have here immunotherapy approaches. So, cancer vaccines targeting TRT derived peptides to simulate cytotoxic T cell responses against telomerase positive tumor cells. So, example, GV1001 or the TRT peptide vaccine. So, it's more on the immune system. So, the big idea here no is since telomerase is inactive in most normal cells, these therapies could specifically target cancer while sparing healthy tissues. So for the clinical relevance and research, okay, as a diagnostic mass marker, telomerase activity can be detected in about 90% of cancers, making it very, very useful for identifying tumors. And as a prognostic marker, certain TERT mutations are associated with more aggressive cancers and poor patient survival. And as a therapeutic target, telomerase inhibition is now at the front line of experimental treatments with drugs and vaccines being tested. So, telomerase is not just a textbook concept, so it's actively shaping how we diagnose and treat cancer. So, to conclude everything, telomerase is a normal enzyme meant to preserve special cells, but when its regulation goes wrong, it becomes one of the most powerful tools that cancer cells use to survive. And by understanding that genes that regulate telomerase Researchers are finding ways to turn this advantage into a weakness and hopefully develop more effective cancer treatments, especially for the future. That's all for my part. Let's move on to the next topic.
All right, thank you so much for that. Can you please send the reactions if I'm audible in your end? All right, thank you so much. So, moving on to the next topic, which is the genes that promote vascularization. All right, so first let's understand what is vascularization means. So vascularization is the biological process through which new blood vessels forms, playing a critical role in embryonic development, tissue repair, and maintaining homeostasis. So basically, vascularization is just forming new blood vessels. So there are two main ways blood vessels are formed. First is vasculogenesis. Next is the angiogenesis. So vasculogenesis, it forms new blood vessels from scratch using precursor cells called angioblasts. And the next is angiogenesis creates new vessels by budding from pre-existing ones. So today, um, we will be focusing more on angiogenesis since it's more connected in cancer. So, angiogenesis, as what I've said earlier, is the development of new blood vessels from pre existing cells. So, it is the process where blood vessels grow from the ones that already exist. So, to imagine this better, think of it as a tree. The big branches are your existing blood vessels, and the small branches sprouting out are the new ones. So that's how angiogenesis works. Okay, so angiogenesis normally happens in healthy ways. Let's say, for example, during growth and development. So, our body forms new vessels to supply growing tissues. And another example is wound healing. So, when we get a cut, angiogenesis brings fresh blood supplies to help the wound repair itself. But here's the problem, not all angiogenesis is good. Sometimes diseases use this process to their advantages. A good example is with tumors. So a tumor is basically an abnormal mass of cells. So not all tumors are cancerous. Okay, so take note of that. Some are benign, meaning they don't spread. But when a tumor is malignant, that's when it becomes a cancer. So cancerous tumors are dangerous because they don't just stay in one place. They grow uncontrollably and can spread to other parts of the body. But for them to do this, they need oxygen and nutrients, just like normal tissues do. So without blood vessels, a tumor can only grow to a tiny size. So this is where angiogenesis comes in. Tumors hijack angiogenesis by tricking the body into growing new blood vessels around them. So these new blood vessels act like a food supply line, giving the tumor oxygen and nutrients. So because of this, the tumor can grow much bigger and can even spread or metastasize. So later on, I will explain what is metastasis to the other organs. So that's why scientists say that tumors cannot grow beyond a certain size or metastasize without angiogenesis. So it is according to Apti et al. 2019. So in short, angiogenesis is one of the reasons why cancer can develop and become life-threatening. All right, so let's move on to the role of vascularization in cancer. So as what I've said earlier, vascularization, specifically the angiogenesis, is a process of forming new blood vessels. And in cancer, this process plays a very big role. Why? Because new blood vessels provides tumor with oxygen. So which are the fuel they need to keep growing? So what I've said earlier, okay? So without this constant supply, tumors would stay very small. But that's not the only role of new blood vessels. They also provide a pathway for metastasis. So metastasis means, take a look at this figure. So metastasis means the spread of cancer cells from the primary cancer or tumor to the other parts of the body. So as you can see here, it transfers to the liver and to the lungs. So going back, you can imagine blood vessels like roads or highways. So once cancer cells enter them, they can travel to new location. This is one of the main reasons why cancer often can backs or recurs and why it becomes harder to treat as it progresses. So cancer cells are very smart in how they do this. They produce large amount of proteins called pro-angiogenic proteins. So these proteins are signals that tells the body, make me more blood vessels. So normally, our body has a balance between signals that promotes angiogenesis and signals that stop it. But in cancer, this balance is disrupted. The pro-angiogenic proteins takes over, leading to uncontrolled blood vessel growth that supports the tumor. So, inherited cancer syndrome, such as von Hippel-Lindau syndrome, has been linked to genes that disrupt angiogenesis control. So, this makes people more prone to developing tumors since the switch for blood vessel growth is not working properly. Now, sorry for that. Now, let's proceed to the genes that promotes vascularization. So first is the vascular endothelial growth factor or VEGF. So VEGF is a central gene family involved in the formation or growth of blood vessels via vasculogenesis or new vessel formation and angiogenesis, growth from existing cells. So it restores oxygen supply to tissues under hypoxic conditions and is crucial for embryonic development, wound healing, and pathological states like cancer and diabetic retinopathy. So here's how it works. So VEGF binds to special receptors on the surface of endothelial cells. And endothelial cells here, as you can see in the figure, so endothelial cells are the ones that line the inside of blood vessels. So one key receptor is called VEGFR or VEGFR or vascular endothelial growth factor receptor, specifically the VEGFR2. So when VEGF attaches to this receptor, it triggers a chain of signals inside the cell. So these signals cause the endothelial cells to multiply, move toward the tumors, and build new tiny blood vessels called capillaries. So in simple terms, VEGF acts like a construction manager. So it tells the endothelial cells where to go, how to grow, and how to connect so that new blood vessels can form around the tumor. But here's the problem. In cancer, the balance is broken. Normally, the body has both promoters and inhibitors of angiogenesis, keeping vessel growth under control. So tumors, however, overexpressed VEGF genes, meaning they produce too much VEGF while the natural inhibitors are reduced. So this imbalance leads to uncontrolled vessel growth. So as a result, the tumor gets a steady supply of oxygen and nutrients, and at the same time, it creates a more escape routes for cancer cells to spread. So this is why VEGF is considered a key target in many cancer therapies in our society. Moving on to the next genes that promote vascularization is the fibroblast growth factor, especially the FGF2. So FGF2 is a general promoter of growth, survival, and motility of endothelial cells. So here is how it works. It binds to special receptors on the surface of cells called FGFRs or fibroblast growth factor receptor. So when FGF2 activate these receptors, it starts a series of signals inside the cell. So these signals help endothelial cells multiply and migrate, which are key steps in forming new blood vessels. So what makes FGF2 even more powerful is that it doesn't just work on its own, okay? So it can also increase the levels of VEGF. So the master signal for angiogenesis that is discussed earlier. So that's VEGF, okay? So by boosting VEGF, FGF2 strengthens the overall process of angiogenesis, making tumors even better at building their own blood supply. That is according to Farouk et al. 2021. So in terms of cancer, this is a big problem. So when FGF signaling is elevated, tumors may use it as a backup plan. Yes, a backup plan. So even if doctors try to block BEGF with anti-VEGF treatments, the tumor can still rely on FGF2 to continue building blood vessels. So this makes cancer harder to treat because the tumors finds another way to survive and grow. So in layman's terms, FGF2 is like a partner in crime for VEGF. Like it helps tumors maintain angiogenesis, even resist some cancer therapies. And moving on to the next gene that promotes vascularization is the platelet-derived growth factor. So PDGF primarily recruits parasites. Or when we say parasite, it is the supportive cells to the newly formed cells, which increases vessel stabilization and reduces leakiness. So its main job is to recruit parasites. When we say parasites, this is the supportive cells that wrap around newly formed blood vessels. Take note of that. So parasites act like a scaffolding around a building. So they act like they stabilize the vessel and prevent it from leaking. So without enough parasites, blood vessels stay immature and fragile. So here's how PDGF works. Here, as you can see in the figure, so tumor-associated endothelial cells release a type of PDGF called PDGFB. So some of this PDGFB binds to receptors on parasites called PDGFR beta. So PDGFR beta. So when PDGFB successfully binds, the parasites attached to the vessels stabilize them. So if PDGFB does not bind properly, the vessels remain unstable and immature. So that is according to RICA 2012. So in terms of cancer, tumor blood vessels are usually incompletely covered with parasites. So which makes the blood vessels abnormal in structure. So despite this, the blood vessels are still functional enough to deliver oxygen and nutrients to the tumor, helping it survive and grow. So in simple terms, PDGF is like the builder that adds support beams to the blood vessels. So when this process is faulty in tumors, the vessels are oddly shaped but still manage to feed the tumor, contributing to cancer progression. So next up is the angiopoietins, specifically the ANG1 and the ANG2. So let's start with ANG1. So ANG1's main job is to strengthen the blood vessels. It does this by activating a receptor on endothelial cells called Tie2, which helps stabilize the blood vessels. So in ANG1, you can just think of this like it stabilizes the blood vessels, right, Jude? So you can think of ANG1 like the construction crew that tightens all the bolts in a bridge, making sure it is strong and steady. So on the other hand, ANG2 has almost the opposite effect. So it loosens the blood vessels. So since ANG1 it strengthens the blood vessels, the ANG2 it loosens the blood vessels, which is completely opposite. So making them more flexible and easier to remodel and sprout new branches. So imagine ANG2 as the crew that temporarily loosens the bolts in a structure so that new extensions can be added. So in terms of cancer, tumors often take advantage of ANG2. So by increasing ANG2 levels, tumors destabilize the vessels around them. So this, combined with VEGF signaling, allows the tumor's blood vessels to branch out rapidly and remain unregulated, giving the tumor more access to oxygen and nutrients. This is according to Actui et al. 2019. So in layman's terms, ANG1 keeps vessels strong and stable, okay? While ANG2 loosens them for growth. So tumors manipulate this balance to help build an irregular but functional network of blood vessels that supports the growth of cancer and its spreading. Moving on. Yes, to the other regulators. First is the HIF1A. It is a gene that senses low oxygen in tumors and boosts VEGF production. Second one is the Notch or DLL4 that helps tumors regulate how many sprouts form, preventing chaotic vessel growth. Third one is the MPP MMPs rather, or the matrix metalloproteinases. So it breaks down surrounding tissue, allowing tumor vessels to push through. All right. So next up, so these are the therapeutic strategies that targets vascularization's genes. So since angiogenesis is essential for tumor growth, blocking of the signals involved with angiogenesis are one of the strategies in cancer therapy. So first is one of the common approach is using the anti-vascular endothelial growth factor drugs or the anti-VEGF drugs. So these are special medicines made up of antibodies like bevacizumab and or VEGF receptor inhibitors. So their job is to stop VEGF and which is the master signal, just like what I've said earlier, for blood vessel growth from working. So it's basically stop the VEGF. So by blocking VEGF, these drugs prevent tumors from forming new blood vessels and slow down their growth. And another strategy is the angiozyme, which is a type of ribozyme. So a ribozyme is a molecule designed to target and destroy specific RNA. So in this case, angiozyme degrades the mRNA for the VEGF receptor, meaning the receptor is not produced. Without the receptor, VEGF cannot send its signals to endothelial cells and angiogenesis is blocked. So this is an example of a genetic approach to stopping blood vessels growth in tumors. So in simple terms, these therapies act like cutting off the tumor supply lines, preventing it from getting the oxygen and nutrients it needs to survive, and blocking angiogenesis is therefore a key strategy is in many modern cancer treatments. So that's all for the genes that promotes vascularization and moving on to the next topic, which is virus infection and cancer.
Am I audible in your end, everyone? Can some reactions to confirm? Am I audible? All right, thank you very much. Now let's move on to the fourth major topic under the genetics of cancer, virus infection and cancer. Next slide, please. Did you know that around 15% of human cancers worldwide are linked to viral infections? Now you do. That means viral infection is actually the second largest risk factor for cancer, next only to smoking. Globally, this accounts for over 1.4 million cancer cases every year. So clearly, viruses play a huge role in the global cancer burden. Next slide, please. So before we move on to these specific cancer-causing viruses, let's start with its history first. However, just to make sure that we'll be on the same ground, let's refresh our memory first and talk about oncogenes, which was tackled already. Oncogenes are the first genes found to trigger cancer. They are altered versions of normal stimulatory genes that, when changed, act in a dominant way to promote the development of cancer. So now that we clearly remember what oncogenes are, let's finally move on to the historical background of virus infections and their relation to cancer cases. Kindly go back. Kindly go back to that slide. Discovery of cancer-causing viruses. All right. So in 1909, Peyton Rose showed that a virus could actually cause tumors in chickens. This was called the Ruse sarcoma virus. His discovery gave us the first strong evidence linking viruses to cancer. Then by 1970, scientists found the first oncogene called SRC from the Rue sarcoma virus. Oncogenes are basically mutated genes that drive cells to divide uncontrollably. Then in 1975, Bishop and Varimus discovered that normal cells actually carry genes similar to oncogenes. These are called proto-oncogenes. Normally, they control cell growth, but when mutated, they become oncogenes and can cause cancer. Next slide, please. So proto-oncogenes can be converted into oncogenes in viruses in several different ways. So first, the sequence of the proto-oncogene may be altered or truncated as it is incorporated into the viral genome. This mutated copy of the gene may then produce an altered protein in the host cell that causes uncontrolled cell proliferation. Alternatively, through recombination, a proto-oncogene may end up next to a viral promoter or enhancer, which causes the gene to be overexpressed in the host cell. And then finally, the function of a proto-oncogene in the host cell may be altered when a virus inserts its own DNA into the gene, disrupting its normal function. All these changes push cells into uncontrolled growth, which is what we call cancer. So while viruses are capable of converting proto-oncogenes into oncogenes, most proto-oncogenes are mutated to form oncogenes without the involvement of a virus. Next slide, please. Now let's move on. Let's look closely at the DNA viruses that are strongly associated with human cancer. First, we have Epstein-Barr virus or EBV. So EBV is best known for causing infectious mononucleosis or kissing disease. But beyond that, it has been linked to several cancers such as Burkitt lymphoma, nasopharyngeal carcinoma, and Hodgkin lymphoma. So how does it cause cancer? EBV infects B lymphocytes, a type of white blood cell, and some epithelial cells. So it carries viral genes that make the infected cells grow uncontrollably. For example, EBV proteins can mimic normal growth signals, preventing the cells from dying when they should. Over years, this constant stimulation can lead to mutations and eventually cancer. Next, we have hepatitis B virus or HBV. So HBV is a major cause of liver cancer. So chronic infection with HBV damages the liver over many years. Repeated cycles of damage, inflammation, and repair cause DNA mutations in liver cells. Additionally, HBV DNA can integrate into the host genome, disrupting normal gene function and potentially activating oncogenes. This is why HBV vaccination is considered one of the most important cancer prevention tools worldwide. Third, we have the hepatitis C virus or HCV. Like HPV, HCV also causes liver cancer, but through a slightly different mechanism. HCV does not integrate into the host DNA. Instead, it causes chronic liver inflammation. Over time, the constant inflammation and scarring create an environment where cancer is more likely to develop. It is also associated with non-Hodgkin lymphoma, likely because of its long-term effects on the immune system and B cells. Fourth, we have the human papillomavirus or HPV types 16 and 18. So HPV is one of the most important cancer-causing viruses. High-risk HPV types 16 and 18 are responsible for about 70% of cervical cancers. They also cause anal, genital, and some oral cancers. So the virus infects the cells of the skin and mucous membranes. Two viral proteins, E6 and E7, disable critical tumor suppressor proteins. So the E6 inactivates P53, which normally repairs DNA damage or causes abnormal cells to self-destruct. E7 inactivates RB protein, which normally prevents excessive cell division. With these breaks disabled, cells grow uncontrollably and cancer can develop. The good news is, HPV vaccines can prevent most of these cancers if given early enough. Next slide, please. Now let's move on to the retroviruses and their associated cancers. So let's first know what are retroviruses. So retroviruses are RNA viruses that use reverse transcriptase to turn their RNA into DNA, which then gets inserted into the host cell's genome. This makes them especially dangerous since they can disrupt important genes permanently. First, we have the HTLV1 or the human T cell lymphotrophic virus. So the HTLV1 is directly linked to adult T cell leukemia or lymphoma. It infects T lymphocytes, a type of white blood cell that plays a role in immunity. The virus encodes the protein called TAX, which drives uncontrolled growth of T cells. So over time, these abnormal cells accumulate mutations and form aggressive leukemias or lymphomas. Second, we have HIV-1, as we all know as human immunodeficiency virus. So HIV is different from other cancer-causing viruses. It doesn't directly transform cells into cancer cells. Instead, HIV destroys CD4 T cells, weakening the immune system. So with weak immunity, the body cannot control other infections or suppress abnormal cell growth. This condition, known as immunodeficiency, allows other viruses and cancers to flourish. So for example, people with HIV are much more likely to develop Kaposi sarcoma, caused by another virus, or non-Hodgkin lymphoma and other blood cancers. So HIV's role in cancer is more indirect. It creates the conditions where cancer-causing viruses or abnormal cells can thrive. Next slide, please. So how do we prevent virus-related cancers? So one of the strongest methods is vaccination. For example, the HPV vaccine can prevent more than 90% of HPV-related cancers, but there is the important part. Vaccines only prevent infection. They cannot treat an infection that already exists. That's why the vaccine is recommended before a person becomes sexually active, since the HPV can be acquired through sexual intercourse. Meaning, prevention is key. Next slide, please. Here are some important reminders. First, viruses can disable our natural defenses. They can produce proteins that interfere with tumor suppressor proteins, the proteins that normally stop abnormal cells or trigger their death. When these are blocked, cancer can form. Second, cancer is a multi step process. So, virus related cancers don't appear overnight. They usually develop years after chronic infection, and viruses are just one factor. Other things like genetic mutations, DNA damage, and failure of tumor suppressor control all combine before cancer actually develops. So the next time we think of cancer, we should also remember that some of it can be prevented simply by protecting ourselves from cancer causing viruses through vaccination, safe practices, and awareness. And that's all for virus infection and cancer.
Alright, so that ends our discussion on the genetics of cancer. So we've learned that cancer is not caused by a single factor, but a combination of genetic mutations and other influences like viruses and the environment. So we also saw how genes involved in DNA repair, telomerase regulation, and vascularization play a big role in cancer growth, and how understanding these can open doors for better treatments and prevention strategies. So we hope these reports gave you a clearer picture of how genetics influences cancer and why studying these processes is so important in the fight against the disease. So thank you for listening.